lot of information, and so we'll start today's right on time. So welcome. When you dial in, you're always just in listen-only mode until the end, and then I'll unmute you. The presentation hopefully will take about an hour. We'll leave a lot of time for questions. The best thing to do is probably write your question down as you think of it, because there's a lot of information today. You can also raise your hand by clicking on the little hand. That way, if I can't unmute you, at least I can read your question and we can get it answered. We're using GoToWebinar software, and so we can put this on the web afterwards. And we are recording the session, so let's get started then. So today's talk is called What We Know About Cancer, but I also kind of call it Cancer 201 because we have a Cancer 101, which is a lighter version of this for people who are just becoming interested in cancer or just becoming an advocate. So Cancer 201 talks about the same things, but we go quite a bit deeper. So CIN's mission, again, is to connect everybody in one way or the other. For me, a real key point in wanting to start a nonprofit was to help people with health literacy, and that's to help them to understand basic research. Most cancer nonprofits don't start that way, but that really is why we started. We wanted people to be able to understand cancer research. That's not only advocates, but cancer patients, the lay community, their family. There isn't a whole lot out there that helps people understand the basic science of cancer and cancer research. So that's one of our primary motives for this nonprofit, and that's why we have such a big site set aside on our website on cancer research, and it's also why we're doing these webinars. So I'm excited about the next couple of webinars, which will really, really focus on the cancer, science of cancer. So all of these webinars are based on the content from our website. So hopefully you were able to go and read it because we're going to move pretty quickly. If you haven't read the section on what we know about cancer, you can go back afterwards and catch up, because every slide has maybe 20 minutes where I could talk about it, but I'll be talking about it for maybe a minute. So if you want to tie the pieces, this webinar sort of is like a 10-story building, and every building has maybe 100 blocks in it, and we're going to talk about 2 or 3 for every level, because that's just all the time we have. So to fill in the key points, go back to that section on the website. It was developed by Diana Chingos, Jane Perlmutter, and myself. And then today's PowerPoint was actually developed by myself with input from Carol Boss. So thank you to all those people who helped provide that content for us. Appreciate your help. So again, welcome, and please ask questions at the end. Write them down. We're again not offering any advice or recommendations, but just providing information. So hopefully by the end of this session, you'll have a better understanding about what we do know about cancer. And of course, that's changing every day, which is very exciting. We'll also become familiar with the basics of cancer biology and the basics of genetics. It's really hard as an advocate to get diagnosed, dive into meetings, and be able to make sense of things. So our learning objectives are to help you know some of those basic biology and genetic concepts. And also to understand the progress in cancer research that we've made over the last 15, 20 years. It's amazing, really, and a lot to know. So here's what we're going to talk about. We'll give a brief overview of what cancer is. Again, we'll focus on some basic cancer biology. again on what causes cancer, and then we'll talk about the advances. These are all taken right from the website, so go back if you'd like. So let's talk about cancer overview. Cancer is one of those words that when people hear it, they get scared. Just hearing that word still has the power to make people afraid. So we're going to try to remedy that a little bit today by helping you understand it a little bit more. Although we won't be talking about specific treatments, we will be talking about the research that is leading to wonderful new treatments. And later on in the series, we will talk about targeted therapy and treatments. Probably, I think it's in October or November. But in this presentation, we're going to discuss the basic science, so you really have a solid understanding of cancer and cancer research. So let's start with the definition of cancer. It's a term for disease in which normal cells become abnormal. They begin to grow out of control. It's one of those, people seem to think cancer is one disease, but it's really not. It's a lot of diseases. It's over 100 different kinds of diseases, and they're all called cancer. And they all have cells that are growing out of control. They all have characteristics that are similar, and we'll talk about that. But each one is different, and the approach to treating it is different. Here's the main types. Carcinomas begin in the tissue, and they're usually named after that tissue. So we have breast, prostate, lung, brain. Those are carcinomas. But we also have sarcomas, and they begin in the bone or connective tissue. We have leukemias, which begin in bone marrow, the blood cancers. We have lymphomas that begin in the immune system. And we have central nervous system cancers. So these are all really different categories. Within each category, there's lots of different types and subtypes. So it's complicated. Here's a cell or an image that I really like. It shows the difference between normal cells and cancer cells. And the pathologist, when they're looking at your biopsy, they're going to be able to see all of those. And essentially, cancer cells don't look at all like normal cells. Their nucleus is much bigger. They divide really quickly. The cell signaling is damaged. There's a lot of differences in a cancer cell from a normal cell that you can just see physically looking under a microscope. So they not only behave differently, but they also look differently. This is a good slide on the impact of cancer, and it's a busy slide. There's a whole lot on here. Essentially, cancer is the second leading cause of deaths by disease in the U.S. It's always neck and neck with heart disease. Nearly 1 of every 4 deaths is from cancer. This year, about 101.6 million Americans will be diagnosed with cancer. That's a huge number. And about 37% of those will die of cancer. And it's usually around certain specific cancers have a higher death rate than others. So just getting a diagnosis of cancer doesn't mean your likelihood is 37%. Some cancers have a high likelihood, so that's why the average ends up being that. But you know, it's still about 1,500 people a day are dying of cancer, and that's about 1 in 2 men sometime over their lifetime, 1 in 3 women sometime over their lifetime. This is all pretty standard traditional information that you can get anywhere, but I liked this little poster because it kind of puts it all together for you. So you can find it on the website, or you can go back to the PDF that I sent and spend a little more time looking at that. So now let's talk a little bit about the basic cancer biology. Biology is a branch of natural sciences that studies the structure, function and growth of all living things. We're going to talk about some of those basics now, but again, we're going to move so quickly that if you want more information, we'll have to go back to our website. So here's what we'll talk about. We'll start with the cell. And this is all stuff that you probably learned a long time ago in high school biology. And you've probably forgotten it since then. And the reason we go through it is because once you understand, especially the cell cycle, it helps to understand what some of the errors are and some of the problems and where some of the mutations occur. And that's why we take you back this far. Understanding cancer genetics is helpful. Knowing the language of biology a little bit will be helpful. We'll go through just a little touch of what causes cancer and a little bit about what genetic variation is. It's really tough as an advocate to go in and sit in a meeting and not have this all tied in. And most of us have been advocates for a long time. And we pull together a little bit from one research meeting and a little bit from talking to somebody and a little bit from reading a protocol. And we try to put it all together in our head. And sometimes we have some gaps. And so hopefully this series will even that out and will fill in some of those gaps where you're not quite sure, why did this happen and why is that there? So that's the purpose today, is to fill in some of those gaps. So let's start with the cell. The cells are the smallest living unit known, and they're usually called the building block of the body. They make up the tissue, which then makes up the organs, and then these organs keep us functioning. And I think that image there is a nice little diagram of how that works. We have trillions of cells in our body, and they're dividing all the time. Each of these cells has its own lifespan. Before it dies, it makes an exact copy of itself, so life goes on, but mistakes can happen during that division process, and we'll talk a little bit more about that in the next couple of slides. So let's talk just a bit about the structure, just so you know what it is. There's some key points. The nucleus is the brain of the cell. That's where everything is housed. The cytoplasm is a gel-like substance that kind of holds everything together. And then there's a membrane around each cell, and that's kind of like our cell or our skin. So those are the main components. All cells have these components. The chromosomes that contain our DNA are located in that cell nucleus, and you can see that from the slide. And again, the cell membrane is surrounding each cell we have. So cell communication, there was a lot of research on this quite a few years ago. And it's because it's so critical that cancer really is a communication failure in the cell. Cells communicate with one another and with themselves, and this is called crosstalk. So each cell is sending signals throughout itself, and that image here, you can see that that's one cell, and if there's a mutation in one of the genes in that cell, it blocks the pathway so that whatever's happening along that pathway can't happen anymore. So it's one of the ways where problems can occur, and that's just a signal in the cell is being blocked. There's also communication between each cell in our body, and that's also called crosstalk, and you can have problems there. So you can have a problem either internally or in cell-to-cell communication. So I think that image kind of helps explain how important that cell communication is. It's something that we forget about. We haven't had pathway talk discussions as often because this was really where cancer research really got its basis, was when people started learning how this worked, and there was lots and lots of pathway research. And so a lot of our basic knowledge is based on this basic understanding. But there's still more to know. There's still more to know, and so there's still researchers who are looking at this. The cell cycle is important. It's a critical process. It's very defined steps. that a cell undergoes in order to copy itself. And it has checkpoints. Each cell has built-in checkpoints that exist in each phase of the cell. Cell division is one of the places where if an error occurs, cancer can develop, or if a cell keeps dividing rather than just dividing once and then dying. If that cell that's divided doesn't die, then you've got too many cells. So the cell cycle is important. In the course of our lifetime, our cells will have divided over 10,000 trillion times. So you can see how that division can result in errors that we just can't correct every time. It happens so fast and so often that sometimes there's mistakes. So here's a nice image of those cell checkpoints. And essentially what that is as the cell is dividing, and remember this goes really fast, but as it's dividing, there will be different phases. There's a G1 phase, an S phase, a G2 phase. At each end of the phase, the cell asks itself, are you okay? Did that work okay? Did everything happen right? And if the answer is yes, then the cell moves on and keeps going to the next phase. But if it doesn't, if some little error occurs right in that G1 phase, then the cell dies. It's programmed to do that. That's called apoptosis, when a cell dies. And it's built in so that to protect you from mutation and cell errors. But because we're making so many divisions and so many cells, sometimes these errors get through. So sometimes the cell ignores the checkpoint, and if the answer is not okay, it still keeps going. So it's one of those points where errors can happen. But the cell cycle and the checkpoints kind of explains how amazing our body really is. We've got lots and lots of built-in protections to keep us healthy, to keep us strong, to keep us cancer-free. And this is one of the key critical ones. As the cell divides, it's always checking to make sure that it's doing it correctly. So all cells are a balance between cell growth, called proliferation. So you probably have heard that word proliferation, and it's just cell growth, or cell division. And cell division can also be just an exact copy of itself, or it can be differentiation, where a cell is changed a little bit along the way. And then there's cell death, called apoptosis. So there's this elaborate system between these three: cell growth, cell death, cell differentiation. All of it has to stay in balance for us to stay healthy. So it's one more little piece of the picture. All of this is going on in the background. We're not aware of any of this. It's just all out there happening. So let's switch a little bit from cells over to cancer genetics. I like little catchphrases. It's all about you. It's very specific. It's very, subtypes are becoming more and more important. Cancer genetics is becoming more and more important. It's important to understand the difference between genetics and genomics. Genetics is the study of one gene and what happens to that one gene in isolation. You can have a mutation and it could be inherited, or you can have a mutation during your lifetime, and then that would be called somatic. But it's one gene and looking at one gene. And there are researchers who look at one gene. Genomics looks at how all of your genes interact with each other. And that interaction can be faulty as well. Research has taken us away from one gene. As we learn more and more about one gene, you then become, have teams of researchers who are looking at this gene and that gene and how they interact and all the rest of it. And that's when genomics came about. And so it's one of the progresses that we've undergone, and it's gained us a lot more new knowledge as we've learned that it's not just one gene that matters, it's how they interact with each other. And that's what the study of genomics is. So this is a really nice image. We'll use it again, but we're using it this time just to explain the 5 basic layers of genetic structure. We've got DNA, we've got genes, there's chromosomes, there's RNA, and there's proteins. So all of these 5 basic players have a role. And we'll talk a little bit, just a little bit about each role, because we're moving quickly again. So again, go back anytime to the website to learn more. But this image here kind of shows you, here's the cell, here's the nucleus, here's the chromosomes in the nucleus. All of them together is your genome. The chromosomes make up the DNA. The DNA then is composed of genes, depending on what the structure is, and those genes then make proteins. So it's a nice picture that puts it all together for you. So let's talk just a little bit about DNA, so that you have a little bit more knowledge of what that is. It's often compared to a blueprint or a recipe for a cooking book, because it tells cells what to do. It's essentially telling them what to do. That's what the DNA does. It's made up of 4 bases: adenine, which is always a capital A; cytosine, which is always a capital C; thymine, which is capital T; and guanine, which is capital G. You have 2 strands of DNA. They're wrapped around each other, called a helix. And A always pairs with T. It can be AT or it can be TA. It can line up either way, but those 2 pair up together. C always pairs up with G. It can be CG or it could be GC. So that's the signature. When someone's doing a sequence, they will be looking at these 4 bases and how they line up. So a little bit about genes. Genes are made of DNA. Before they sequenced our genome, they thought there were 100,000 genes. People didn't really know, but they guessed big. They found out there's really only about 20 or 25,000 genes. There aren't near as many as they thought we had. I think the key point for this slide is to know that genes make proteins. And proteins then act as the instructions telling our cells what to do. We know that every person has 2 copies of each gene, 1 from each parent. Here again is a picture of a chromosome, tightly wound up. Most genes are the same in all people. 99.9% are the same. But a small number are slightly different. It's what makes you different from the next person. Some genes are turned on and function, and others are turned off and don't function. And we'll talk a little bit more about that as we move through it. So we're sort of building a case. We're building the vocabulary of this basic biology and genetics. So the chromosome is this really thin thread-like structure of DNA, and it carries the genes. That's what it is. Each chromosome is made really tightly coiled. There's about 25,000 different genes. Again, we talked about the fact that we have about 20 to 25,000 different ones. We have 22 pairs of identical chromosomes, and 1 pair that's different. The sex chromosome, which is XX, an X from mom and an X from dad, and the male, which is an X from mom and a Y from dad. So those are our chromosomes, so 23 pairs. RNA isn't talked about quite as much, but it is the structure that actually transfers the information. There's a central dogma, which Francis Crick wrote out years and years ago. DNA makes RNA, RNA makes protein, and protein makes you, makes us. It makes us who we are. Again, we're all pretty close together in our DNA sequence with tiny little changes, but that's how it works through, that we have DNA, which then makes RNA, RNA makes protein, and proteins make us. So what are proteins? Remember, they're made by genes. They regulate all our body functions. Some example of proteins are hormones, or enzymes, or antibodies. When a cell needs to make a specific protein, that gene switches on. And a long, complicated process occurs that involves both DNA and RNA and results in a protein being made. And we'll talk a little bit more about that, but there's a lot more about that on the website. To give a 1-hour talk that covers this broad a topic, all I'm really giving you is some vocabulary and a little bit of if this were an arrow moving through, so that you're kind of building the vocabulary of cancer research. So gene expression, it's one of those terms that you'll hear in a research talk, and the definition is the process by which a gene gets turned on in a cell to make RNA and proteins. And that journey from gene to protein is very, very complex. It's very tightly controlled. There is a picture of it. It can have errors along the way. It's another place where problems can occur. First you have transcription and then translation. So again, we're just defining the term for you, letting you know that when the gene gets turned on, that's when you use the term gene expression. Genes sit there all the time, sometimes not functioning. It's only when a gene is turned on that it begins to function, and it makes a protein and something happens. So just, we took about 10 minutes to go through this really huge topic. Essentially, cancer is a communication failure in the cell, and we saw a good image of that, of how that can happen. We talked a little bit about DNA, that there's this delicate balance between proliferation and differentiation and apoptosis. That's happening in the cell cycle. We talked a little about genes making proteins, and that you can have inherited mutations, although very few, and somatic mutations. Those are mutations that occur in your lifetime, and that's where most of mutations actually happen, is during your own lifetime. And the term that gene expression. So that's a quick little summary of what we just talked about. Now we're going to move into what causes cancer. It's essentially a renegade system of growth that originates within someone. There are many different kinds of cancer, but they all share this one characteristic of unchecked cell growth that may progress toward limitless expression and expansion. And we'll discuss this process in the next couple of slides. As you can see here, we have a couple of causes right here, but we'll go in and talk about this in a detailed way. So two things have to happen for you to have cancer. You either have DNA damage, which is a mutation, and that can happen during the cell cycle, when the cell is copying itself. It can happen because of a communication error, and we saw slides on that. It could happen because of carcinogens, chemicals outside us that can cause damage. Lifestyle, they found, has a role. Aging has one of the biggest roles. Cancer occurs as you get older, much, much, much more frequently. And part of that is just because our body isn't as good at fixing the errors during that cell cycle, cell copy phase. Some of the repair genes aren't functioning as good. As you get older, things don't work as good. So aging is one of the primary causes for cancer, just because all the mechanisms in place to keep you from getting cancer don't work quite as well. Radiation has been determined to be a cause of cancer, and viruses and other environmental exposures. So we'll talk a little bit more about that in our next slides. And recently, we also learned that epigenetic damage can also be a cause. And in fact, almost all cancers have both DNA damage mutation and epigenetic damage. And what that is it's damage to the environment around the gene. The gene isn't mutated. The gene is okay, but something around the gene has changed. And it's caused that gene to be silenced and not to function well anymore. So although it's not mutated, it's not functioning, and it's not producing the protein that it should be. So both of those things have to be in play for you to get cancer. So a little bit more about DNA damage. It's the hereditary mutations. It's only about 10% of the cancer, and those come in germline, and they're inherited. Most people seem to think that more cancers are inherited than are. And although there's research that there may be other genes involved, and we are finding that is a bigger field, that we know of some genes, but they're always finding other little ones that may be involved as well. So although right now they're saying about 10% of cancers, I think that's one of those areas that we'll continue to learn more about, and more information will be found. Maybe there are more genes that are causing cancer that they just haven't found that are inherited. But today they think most of the mutations occur during your lifetime. That's called somatic. And at least today they're saying that most cancers occur somatically. Genetic variation is interesting, and we'll talk a little bit about that so that you can get a little bit of understanding of what that is. It's actually just a difference in the sequence. Remember, we talked about A, C, G, and T. That's the DNA sequence. Those are the 4 bases, and if those sequences get out of alignment, you can get a little bit of genetic variation. And that's the occasional single letter difference that distinguishes DNA among people is called single nucleotide polymorphism, or SNP. And SNPs are the most common type of genetic variation among people. And each SNP represents a difference in just one single DNA building block, but it leads to difference in blood group, eye, skin, and hair color, higher or lower risk for disease. It explains a lot of the differences in us, and it's just in that little DNA sequence. So let's take a little bit more look at that, what that SNP is. So you can see from the image that here's a DNA sequence. That's T, A, G, and C. That's the common sequence for this gene, and it makes a specific protein. If something happens to that sequence and it now becomes TACC, then that's called a SNP. It's one single nucleotide has changed. There's a variation. It's called a SNP, and the protein it produces is different. Now, maybe that matters, and maybe it doesn't. It depends on the protein, depends on the gene. So there's a lot of research into SNPs, looking at precancerous conditions, looking at drug resistance, looking at cancer susceptibility, and drug response. A lot of these SNPs are responsible for these areas. And so there's quite a bit of research looking into SNPs now, but that's essentially what it is. It's one single letter in this sequence that has been changed. And to be called a SNP, at least 1% of the population has to have that. So if you were to look through everybody, you would have to see that in at least 1% of the population for that to be identified as a SNP. Otherwise, it could just be an error. You know, errors happen all of the time. But if it happens in a large percent, 1%, that's a lot of people. So this same error occurs in 1% of the people. Then it's called a SNP, and it may affect all of these areas. And so there's research into identifying SNPs and identifying ways to treat that particular sequence problem. So let's do a little bit of a summary now on cancer biology. Again, we just whizzed right through that, I'm sorry to say. But damage to DNA can be caused through environmental triggers. It could happen in cell cycle division. It could happen because of infectious agents and viruses, aging and lifestyle. We can have genetic mutations that are inherited or acquired. And the changes can occur in the gene, in the chromosome, and in your cell. So any place along that line, you can have some problem. Epigenetic damage around a gene can also cause cancer, and that's one of the newest areas. I mean the last 15 years or so. People didn't really believe that for a long time, and we'll talk a little bit more about that later. So let's go on to advances in cancer research, because that really is an important and exciting new area. So here's the overview. The 6 hallmarks of cancer. It was an important paper written in 2000. You can get this paper anytime, just Google it. Just the 6 hallmarks of cancer. We'll also discuss some of the key genes involved in cancer, and those are oncogenes, tumor suppressor genes, and DNA repair genes. Each of those genes is important. There are lots of other genes that are involved, but these are the key ones that we've known about for the longest. And then we'll talk about some recent research hallmarks that weren't mentioned in the hallmarks of cancer paper. So I don't know how many of you have read this paper. This is the image that was designed to help explain this paper, and essentially the authors are saying that there's a group of 6 things or hallmarks that have to happen for cancer to occur. They describe how tumor progression proceeds via this process in which each genetic change confers a growth advantage to the cell. And these build up, and they are grouped into 6 hallmarks, and they drive a population of normal cells to become cancer. So that's what they said. It's held up over all of these years. This picture is widely produced. You've probably seen it someplace. We're going to discuss each of these hallmarks in the next couple slides. One of them is self-sufficiency in growth signals, insensitivity to anti-growth signals, evading apoptosis. We've talked a little bit about how cells are programmed to die. A cancer cell is very good at evading that. Limitless replicative potential. One of the things that people know is that cancer cells are growing out of control. That's limitless replicative potential. It just keeps replicating and replicating. You have tissue invasion and metastasis. That's a hallmark. It does cancer. That has to occur. So let's talk about each one a little more. Um, 4 of these hallmarks occur inside inside the cell itself. And the first one is self-sufficiency and growth signals. Cancer cells that grow even when they're told not to, that's what that means. Remember, we talked about the cell cycle, and we talked about checkpoints, and we talked about how cells will be told, stop, there's a mutation, stop, and that cell's supposed to die. But cancer cells grow even when they're told not to. So they miss that whole stop checkpoint, and they gain the ability to keep growing. Let's go back. There's insensitivity to anti-growth signals. So they don't listen to the stop one. So there's some that are growing even when they're told not to, and there's some that are growing even though they have been told to stop. So those are 2 of the growth ones that are in play, and they both have to be there. There's also evading apoptosis, where they totally ignore the signals to die. So, because remember, during that cell cycle and cell division and the cell communication thing, the cells are sending signals all the time on how to behave, how to be normal. And then the limitless replicative potential, where it just keeps copying itself again and again and again. Those all occur within the cell itself. There's a couple things that happen outside the cell. One of those is the sustained angiogenesis, and that's the creation of blood supply. Cancer can't maintain itself if it doesn't have a good blood supply. So sustained angiogenesis is critical. Without an adequate blood supply to bring nutrients, a tumor can't continue to grow. So scientists have found that tumor cells actually send out signals that encourage the growth of new blood vessels surrounding that tumor. They send out VEGF production, and it says, build new blood cells, build new blood vessels. And that helps that cancer continue to grow, because it needs the nutrients to continue to grow. So cancer cells have that ability. Some primary tumor cells acquire the ability to undergo invasion of metastasis, whereby they leave their original location and they move into surrounding tissue. And then they eventually travel to distant sites. They form new colonies, secondary tumors, and really it's almost always the metastasis rather than the primary tumor which causes death. So this ability is one of the key research areas of focus. What happens? Why do some cancer cells have that ability? How can we prevent that? How can we target that? So this is a huge area of investigation, because if they could figure this part out, then way fewer people would actually die of cancer. People would have surgery, people would have some local therapy, and that would be the end of it. It's only when those cells move out throughout the body and get to places that can't be reached and are so small and they continue to grow that becomes a real problem. So this is a huge area of research, is tissue invasion. So here's just a little bit about the 3 important genes that we discussed earlier. The name of the genes are oncogene, tumor suppressor, and DNA. An oncogene, when a mutation happens in an oncogene, you get uncontrolled growth. And a couple of examples of those oncogenes are HER2/neu, RAS, MYC, and SERP. So we don't have many oncogenes in our body. And so that's a good thing. Another gene that's important is called a tumor suppressor gene. And again, if that gets mutated, you have uncontrolled growth. Um, P53 is an example. RB and APC are other examples. DNA repair genes are the name of another important gene, and a mutation there causes the inability to correct cell errors, which is an important part of any cell, is to correct errors that occur within it as it divides. So BRCA1 and 2 are examples. Many cancers have either mutations or epigenetic changes in all three of these genes. They could all be in play. So finding treatments that correct each of them is difficult, and it helps explain how complicated treating cancer is, because each of these is a very different problem. And if your particular cancer has mutations in all three of these genes, it's going to be tough to treat. So it does help explain why treating cancer is so important. Remember, we get a gene from mom and dad. We have two. So if you have a mutation in one gene, in an oncogene, you're still okay. You have to have that second mutation for the gene to be totally mutated. So we do have some built-in things to help keep us healthy. Again, the body is just a master at keeping us healthy. So you do have to have a mutation in both copies for these not to work. So let's talk just a little bit more about the oncogene. It is a bad guy in the scenario, and it's once it's mutated, it gains the ability to drive nonstop growth. And you can see this little car illustration that when an oncogene arises in normal cells, they contribute to the development of cancer by telling cells to make proteins that stimulate excessive cell growth and division. They're not usually involved in inherited forms of cancer. Most of them occur as somatic mutations, and they can't be passed on from parent to child. So that's a good thing to know. But you can see, once it's mutated, it's pushing cell division. That's what an oncogene does. The normal copy of that gene is called proto-oncogene. So every once in a while, that word will pop up, and it took me, I think I was an advocate for 10 or 12 years before I figured out what in the heck a proto-oncogene was and what's the difference between the oncogene and the proto-oncogene. So the proto-oncogene is the normal copy of that gene. Once it's mutated, they then call it an oncogene. And who knows how any of that occurred, but that's what that's about. So tumor suppressor genes are actually the good guys. You want really well-functioning tumor suppressor genes because they act as a brake during the cell cycle to stop or to slow growth, or if there's a problem. So if there's a mutation in that normal braking mechanism, then you don't have that fix. So tumor suppressor gene is one of those fix spots where, just like in cell cycle, it's stopping and checking all the time to fix problems. Well, tumor suppressor genes do the same thing. If there's a mutation out there, a cancer cell out there, it will go and attack that, and its job is to get rid of that. It's forcing those genes to stop. So when there's a mutation in that and it's no longer available, that's a real problem. Tumor suppressor genes can be inherited, but remember, we get one gene from each parent, so it's rare to get a mutated copy from each parent. Cells that have mutated and are growing out of control can lead to cancer. So that's what the job of this tumor suppressor gene is to stop that process. Same with DNA repair genes. again, they're more the good guys. Um, and remember, if the rate of DNA damage exceeds the capacity of the cell to repair it, the accumulation of errors can overwhelm the cell and result in cancer. So you can see that there's up to 500,000 DNA modification events per cell every single day. Its job is, every time something happens and there's an error, its job is to fix that error. And it does. You can see 500,000 times per cell per day. It's fixing a lot of problems. A lot of things can go wrong. This gene, its job is to fix all of that. If this gene gets mutated and can't do that, then there's no way for those problems to get fixed, and they just accumulate and accumulate, and pretty soon you do have a problem. So that's why it's really important to have functioning DNA repair genes. And this is something they didn't know for a while, that there are actually genes out there that are repairing all of this damage. So there is some research into this. Usually it's colon cancer that has a problem with this. Genes can be inherited. DNA repair genes can be inherited, mutations, and it usually is in colon cancer, but some breast and ovarian cancer as well. So here's, let's move on a little bit to some recent research findings. For me, this is a really, really exciting area, because for the first time, infectious agents are really being looked at. We'll talk a little bit more about that. There's been way more advances in tumor genetics. We have an understanding now of stem cells and what role they may play. We're finding lots more out about epigenetics, a lot more about the microenvironment. We're also understanding the role of the immune system. Understanding drug metabolism really has nothing to do with the cause or the formation of cancer. The only reason I included it here is because most people with cancer are treated with a drug. And recently, in the last 10 years or so, we found that how people respond to that treatment is different. And that depends on their drug metabolism. So we're going to talk just a little bit about that, just because it's something important to know. So infectious agents, it was shocking to me to learn that close to 20% of all cancers are caused by infectious agents, such as viruses and bacteria. And there are some people who think much more, than a much higher rate than 20%. A lot more cancers may be caused by infectious agents, that they just haven't found that causative agent yet. But there are people looking. We know that at least 20% are. Some of the examples are HPV, which leads to cervical or esophageal cancer. Epstein-Barr has a role in lymphoma. Hepatitis B with liver cancer. HTLV1 with acute lymphocytic leukemia, and Heliobacter pylori with stomach cancer. One of the first things you need to help people understand if you're talking about infectious agents is that you can get the virus, you can get an HPV virus and not get cancer, or Epstein-Barr and not get cancer. Something else has to occur in your body. So cancer is not contagious like the virus is contagious. And I guess the best way to explain it is like in your family. If someone in your family has the flu, and there's 5 people in your family, and the flu virus is all over your house, everybody's ingested it, they all have the flu virus, but maybe only one person comes down with the flu, because the other 4 people, their body has taken care of that flu virus. It's counteracted it, and it's no longer a problem. And so the same thing with the virus. You can get one of these viruses. It is contagious, but that doesn't mean that you will get cancer, and it doesn't mean that you can spread that cancer. It just means that it has a causative role, and maybe 4 or 5 or 6 of these other, all these hallmarks that we talked about, those things have to happen as well. But it's important to know that there are agents out there that can cause cancer, and so it's a big area of research. It potentially can lead to vaccines and ways to treat cancer once you know what that causative agent is. It certainly is an exciting area, and in some parts of the body or some parts of the world that have more of these viruses, that their 40% of cancer may be caused by an infectious agent. So it's that 20% is just here in the U.S. So tumor genetics, we've learned a whole lot more since that 2000 paper, that Weinberg paper. It takes several different types of mutations to contribute to malignant changes. We know that there are other mutated genes besides oncogenes, tumor suppressor, and repair genes. We've found a whole bunch more genes, and I'm sure there are more yet to find. We do know that most cancers are sporadic. There's a whole lot of information that we've learned new in this area, and there's people that's what their focus is. Cancer stem cells, this is still an area that has some controversy around it. There are people who believe in the stem cell hypothesis, and that's that there's a small portion of cancerous tumors that are made up of stem cells which are resistant to current therapies. That you can treat the tumor, but if you don't kill those stem cells that are in the tumor, they'll come back. Maybe it's 5 years, maybe it's 10 years, but they'll come back, and it's because you haven't killed that stem cell. There are other researchers who don't believe that. So this is an area of active research. There are actually therapies out there that are targeting cancer stem cells, and that will provide new information if they work. And maybe there's, just like everything else, maybe there are some cancers where this is in play. Maybe there's some people where this is in play. It's still an area that's undergoing more research. So epigenetics, it's an area I just really like. I think this is a really important area. Remember, it's that area outside the gene. It's not the DNA. It's around the gene, and somehow that gene is silenced. Methylation is one way that can happen. It's an example of epigenetic change. The methyl group gets attached, and it can silence that gene. And again, most cancers are a mixture, and they found that actually there's way more epigenetic changes that cause cancer than there are genetic changes. And this was shocking to those two people who have focused their whole life on genetics, to learn that there are other things going on. And that's in the microenvironment. That's the extracellular matrix. And it's a network of proteins, and it surrounds the cells. It provides structural support. It's a communication pathway. There's identified as key for normal functioning of cells, and if you have a problem in the extracellular matrix, this can lead to cancer. It's one of those areas that people didn't believe for a long time. I don't know if any of you know Mina Bissell. She's from Berkeley. She presented this way back in the 70s. People thought she was crazy. Essentially, she was saying that this was so. And she even went so far as to say that the tumor microenvironment can trigger cancer. That if something's wrong in the microenvironment, it can trigger the cells to mutate. So that's